Okay, so this is a walkthrough of the revision questions that I set you. I'm just going to go through the answers and you follow along and correct them at home. So question 1, you have a picture of some cells in the lining of the stomach and you're asked to identify the structures A and B. Now cells weren't part of this topic, however this is an example of what we call a synoptic question, where the question may be about one topic but they would mix in another topic just to make sure that you are keeping up with the other topics. So A, it's the outside, and it's a stomach, so these are going to be animal cells. So A is going to be our cell membrane, and then B, it's the inside of the cell, that's the cytoplasm. What is the function of the nucleus? You've got 3 options, and it controls the activities of the cell. Movement of substance in and out, that's the membrane, and release energy and respiration, that's mitochondria. You don't have to do these to get the mark. I'm just going through how I would do the question by double checking by eliminating the other answers. It's a good tip if you can do it. B, draw one line from each part of the human body to its correct scientific name. Okay, so you've got layer of cells lining the stomach, and the mouth, stomach, intestines, liver, and pancreas. Well, I'll start with the ones that I think are the easiest, and that is that the stomach is an example of an organ. Mouth, stomach, intestines, liver, and pancreas, organ system. Now this one, a layer of cells lining the stomach, so that's cells together doing a job together, tissue. When you're drawing lines, I know it seems obvious and a bit silly, but make sure that your lines are actually touching the boxes. Some people draw lines like that, and if you get a particularly picky examiner, that wouldn't count. So also that's wrong, but make sure you're drawing your lines correctly. Okay, on to question 2. Diagram shows a section through the heart. Oh no, I keep pulling it down to be nearer to me, but then it goes out of the camera. Use the words from the box to label parts A, B, C, and D. Now, you know slightly more, or you should know slightly more accurate names, but this example we've been given the names and we have to fit them in. Okay, so A is coming out there, that is an artery. If you didn't have a box of words to pick from, that would be your aorta. B then is a ventricle. We know that to be the left side, because first thing, this is my right hand, and then back to front, because it's if you're looking at it in someone else. So this is the left side and the right side. Also, you can kind of see here a little bit of a thicker wall. That's the left side is going to the body, so it needs a bigger push. C then is an atrium, specifically the right one, and D is a vein. Proper name, vena cava. Now, based on the mark scheme, if you had put the more complicated names that you had known, you would not have got this wrong, even though they asked you to put the ones from the boxes, because you're showing that you know it's a vein, it's this vein, and this is the name of it. So don't worry about that. Okay, on to diagram 2, which shows your treatment for a diseased coronary artery. So it shows you the coronary artery on the outside of the heart, and it shows you a picture and says that there's a wire mesh inside that coronary artery. Name the treatment shown in diagram 2. Well, the wire mesh, that is a stent. And explain how the treatment works. 2 marks, so you've got to say 2 separate things. First thing that you need to say is that it keeps or holds the artery open. Or you can say things like it pushes the blockage away if it was blocked with cholesterol or plaque. And then, so that's the first point. The second mark is why that's useful. How does it work? That allows the blood to flow easier, or so more blood can flow. If you've written things that are similar, but you're not exactly sure if you said the right thing, you can write it in a comment to me or take a picture of it and send it to me, and I'll double check if there's some things that you're not entirely sure on. Okay, question 3a. Name the organ which makes bile. That's your liver. And label this organ with the letter X on the diagram. So you need to write on the diagram. Now, you wouldn't usually be able to do this on a test from school, but on your real GCSE papers, you of course can write on them. So the liver, I'm going to shade in here. Like that. So that's the liver. The advice in the mark scheme says that it's got to be very, very clear that it's on that pit and it's not overlapping on this little organ here, which is the gallbladder. So it must be clearly just on the liver, not overlapping any other organs. Information in the table may help you to answer parts B and C. Name two parts of the digestive system where protein is digested. So you're looking for anywhere where the enzyme that digests protein. And if you look here, we've got stomach, we've also got small intestine, and we've also got pancreas. But the reason why this one is not correct is because food doesn't actually go into the pancreas. The pancreas releases the protease into the small intestine. So two answers are stomach and small intestine. So just two reasons why starch is not digested in the stomach. So stomach is there. Starch is digested by the enzyme amylase. So you're looking for the locations of amylase. So your first mark is to say, well, there's no amylase made in the stomach. Your other mark is in this column, and that it looks like amylase only works between sort of 7 and 8.5 pH, whereas the stomach is really acidic at pH of 2. So the second mark for part C is to mention that something about it being too acidic or the wrong pH, or that the enzyme, our favorite word when we're talking about enzymes, would be denatured. Or there's a lot of options in the mark scheme that it only works in neutral or alkaline pHs. So there's quite a few options there for that second mark. The first one is just to state that, well, there's no enzyme made. There's no amylase made in the stomach. Okay, on to question 4. Now this question gives you a little bit of background about an experiment that some students are doing. When you get a question like this, don't be tempted to skip over and just start answering the questions, because you might be missing some key information and end up getting an answer wrong just because you haven't read through it. So they're investigating the effective temperature on the action of the enzyme lipase. They put 1 centimeter cubed of lipase into a test tube, demonstrated here, 5 centimeters cubed of lipid into a different test tube, there, and put both tubes in a water bath at 5 degrees for 3 minutes. Then they mixed the lipid with the lipase solution, there. Every 5 minutes, the student tested the sample of the mixture for lipid until no lipid remained. The students repeated the experiment at different temperatures. To make their investigation fair, the students needed to control some variables. Give one variable the students controlled in their investigation. That's what I've got the highlighter for, because I'm going to go and highlight the various bits. So here is something that's a control. They used the same volume, not amount, the same volume of lipase. They also used the same volume of lipid, and they left them before mixing them for the same amount of time. So any 3 of those answers are correct. A common incorrect answer here is temperature, but they're not controlling temperature because it says that the experiment is being done at different temperatures. So that's not a control variable, that's the independent, the one that's being changed. So the volume of lipase, the volume of lipid, or the 3 minutes before mixing. B. The tubes of lipase solution and lipid were kept separately in the water bath for 3 minutes before mixing. Why? Well, that is so that they both reached the right temperature. The lipase isn't going to break down the lipid because they haven't been mixed yet, so that's why that's incorrect. To give enough time for the lipase to break down the lipid, again, they haven't been mixed, so that can't happen. To give enough time for the water bath to heat up. Well, there's a few things I can see wrong with that. 5 degrees is actually quite cold, so I don't think it's going to heat up as such. So that's why I'm going to eliminate that answer. Oh, no, next page. Table shows the students' results. Describe the effect on the breakdown of lipid of increasing the temperature from 5 degrees to 50 degrees. So it just wants this bit of the table. So we're ignoring 95 for now. I'm assuming that's, yeah, there's a question on that later. Describe the effect, 2 marks. So you don't have to explain why the times are changing, just what is happening to those times. 2 marks. So you can say sort of 2 different things, and I'd say between 5 and 35. That's your first thing to talk about. So between 5 degrees to 35 degrees, the breakdown speeds up or takes less time. Whereas between 35 and 50, what happens is that it slows down, or to get the second mark in a different way, you could give the optimum. So there's a third option for getting that second mark, and the optimum is 35 degrees C, based on our results. Suggest 2 ways in which the students could have improved their investigation. Use information from the student's method and the results to help you. Well, if you go back to the method, sorry, that's getting a bit wonky. It says that they tested the sample of the mixture to check for lipids every 5 minutes. That's quite a long interval to leave it to see if the lipid has digested. So the first thing, no, wrong sheet, is they could test it more frequently than 5 minutes. Something else they could do, there's quite a big jump here in temperatures. So we go from 5 degrees to 20 to 35 to 50. We get a general pattern, but it's hard to pinpoint exactly where our optimum might be. So we could use more temperature intervals. Now what I mean by intervals is just the gaps. So we could test every 5 degrees, so 5, 10, 15, 20, 25, etc. They could test values between 50 to 95, or, as with any experiment, repeating at each temperature, and you repeat so that you can get a mean value, because it looks like they've only done it once, so that could have been a fluke, it could have been by chance, whereas if you repeat each temperature and get a mean result, you're then more likely to get something that's reflective, that's representative of what's actually happening. Part 1, the lipase did not break down the lipid at 95 degrees. Why? 1 mark. And that's where you need to say that it's been denatured. Or you can explain what denatured means, and that's that the active site has changed shape. If this question was worth more than 1, so for example 2 marks, you'd need to say both of these things. The keyword denatured and the explanation, but it's only 1, so either is fine. The last part, at 35 degrees, the lipase broke down the lipids after 5 minutes. What new substances will be in the tube? So this is what's made when you break down fats, and you get fats, fatty acids, and glycerols. Okay, now for question 5. The photograph shows a red blood cell in part of a blood clot. Fibres labelled X are produced in the early stages of the clotting process. Suggest how the fibres labelled X help the blood clot to form. So if you go back to the question, have a look at the page and the photograph, you've got your red blood cell here, and then the sort of fibres forming a kind of barrier behind it. And now this is a suggest question, so this is not one that you necessarily know off the top of your head. It's one where you use the information given to you to give a best answer that you can. So some idea of holding the cells or stopping them from flowing or from moving or trapping them. The average diameter of a real red blood cell is 0.008 millimeters. On the photograph, the diameter of the red blood cell is 100 millimeters. Use the formula to calculate the magnification of the photograph. And the formula is as follows. Diameter on a photograph is real diameter times magnification. So you don't actually use this equation because you want magnification, but it's not on its own in the equation. So either you can rearrange to do that, or you can use the one that we've learned, which is MIA. Okay, magnification is image size over actual. So the image size is 100. The actual or real size is 0.008. You substitute that in, so 100 over 0.008, which is the same as 100 divided by 0.008. You always get to use a calculator in science, so it gives you 12,500. Yes. They were both already in millimeters, so we didn't have to worry about converting, but quite a common thing with a magnification calculation is that they may be in different measurements and you might have to convert first. Some blood capillaries have an internal diameter of approximately 0.01 millimeters. Use information given in part B to explain why only one red blood cell at a time can pass through a capillary. So 0.01 millimeters, that's how big a capillary is, and our red blood cell is 0.008. So those two sizes are very, very similar. So red blood cell and capillary are almost the same size, or no more room for more red blood cells, or the idea that only one can fit. Through. So they're almost the same size, there's no more room for any more, only one can fit through at one time. Explain the advantage of red blood cells passing through a capillary one at a time. So why is having one cell through at a time good or useful? 3 marks here. So the first thing is about what the red blood cell is likely to be carrying. So if it's going through slowly, it depends where it's going through. So for example, if the capillary is in the lungs, it allows it to take up more oxygen, or if it's been to the lungs and it's delivering oxygen somewhere else, it can release more oxygen. So those are the same point, just the opposite way around. We need to say 2 more things. So why does it going through one at a time allow it to pick up more or release more oxygen? And that's because there may be more time because they're going through one at a time, or it's a slower flow because they're going through one at a time. Because they're only going through one at a time, the distance that the oxygen is traveling is small, or that there is more surface area exposed. So if they're going through one at a time, all of the red blood cell is available for the oxygen to come in and out, depending on if it's the lungs or somewhere else. Whereas if they're coming through lots, some might block bits of others, so it couldn't get as much oxygen. So there's 3 marks there. 1 for saying something about more, releasing more, picking up more oxygen, and then 2 of these. So more time, or they're going slower, the distance the oxygen has to go is small, there's more surface area exposed. Okay, on to our last question. So it's another experiment, and again it's another experiment involving lipids and lipase by the looks of it. The diagram shows the apparatus used to investigate the digestion of milk fat by an enzyme. Oh, I might have given you an answer there. The reaction mixture contained milk, sodium carbonate solution, an alkali, and the enzyme. Experiment 1, bile was also added. Experiment 2, an equal volume of water replaced the bile. In each experiment, the pH was recorded at 2-minute intervals. So you've got 2 experiments, one with milk, sodium carbonate, which makes it alkali, bile, and the enzyme. One with milk, sodium carbonate, water instead of bile, and the enzyme. We've got some results given to us in a table. It says milk fat is a type of lipid. Give the name of the enzyme which catalyzes the breakdown of lipids. If you were listening, I've already told you. Lipase. What was produced in each experiment to cause the fall in pH? So if you look at both examples, they both started out at a pH of 9, and gradually they got less. Now if you think about what pH numbers mean, if the numbers are going down, they're getting more acidic. We're breaking down fats. And when we break down fats, we make two things. We make glycerol and fatty acids. So as we break it down, we're making acids, which is meaning that the pH is decreasing in our experiment. For experiment 1, calculate the average rate of fall of pH per minute between 4 minutes and 8 minutes. Show clearly how you work out your final answer. So experiment 1, this one, between 4 minutes and 8 minutes. So in this time, we go from 8.7 to 7.7. So 8.7 take away 7.7, it gives us 1. So in that time, the pH fell by 1 pH unit. But it wants it per minute. And we've had a period of time of 4 minutes here. So 1 divided by 4 gives you 0.25 pH units per minute. Last question, why was the fall in pH faster when bile was present? 1 mark, you have to give some reference to what bile does when it comes to fat digestion, and that's because it emulsifies or emulsified the fats. Or you could give an explanation. If you can't remember the word emulsify, you can give an explanation of what that means. What that emulsify means is that it takes large fat drops, breaks them down to small fat drops, or it gives it a larger surface area for the breakdown. Okay, I hope that helped. If you have any questions or anything you want me to check, just leave me a comment on Show My Homework.